set up the Plant Biotechnology Laboratory here in West Kingston to work on the introduction and modification of genes that would improve cereal crops, specifically rice, corn, and more recently switchgrass, which were involved with the genetic improvement of switchgrass specifically for biofuels. Yeah, we have a whole team at the Plant Biotechnology Lab here, all of which are specialized in what they do. For example, we have people that are focusing on the techniques to move genes into switchgrass that will improve switchgrass for biomass production. This is transgenic biology, and it's very highly specialized. We're one of very few labs in the world that have this capability down to a routine procedure. And so we also have a group of people that are developing the molecular constructs that we will move in to switchgrass. We have people that are involved with genomics and sequencing the switchgrass genome, which provides us with an important database for genes, but also for doing advanced breeding that will be involved with developing new hybrids. Lastly, we have molecular analysis. Once we move genes in, we have to be able to analyze that outcome.
My job runs a pretty wide gamut of responsibilities. I'm involved in nearly every transformation project that's ongoing in the lab. And that ranges from maintenance of agrobacterium strains and transformation vectors out to regeneration and maintenance of the actual transgenic plants produced, and then subsequently the molecular analysis of those plants.
We're testing a male sterility strategy, nuclear male sterility strategy in switchgrass. The idea is that we would come up with a male sterile phenotype which could be used for gene confinement strategies and also to provide a platform for hybrid plant development. We have a construct we developed which uses some endogenous genes from rice that are tapetum-specific promoters, strictly expressing certain proteins in the tapetum, which is a layer of cells that surrounds the pollen during pollen development. And it allows for us to have tissue-specific expression during that development in the plant. And the idea is that we would induce expression of a cytotoxic Barnase gene, which is an RNase, which would at that point degrade all RNA within the cell, ablating all pollen, and lead to a male sterile phenotype.
My job as a research assistant and a PhD candidate in this lab is to study some underlying genetic mechanisms that are involved in sex determination in co-sexual cereal flowers, and see how that information can be extended to produce male or female sterile co-sexual cereal crops for the use of hybrid breeding. So this could also be used, the hybrid breeding technologies that we make, that we can develop, can be used for biofuel research.
Well, we realize now that as alternative fuels become a possibility in the United States, that the resources that are required to make liquid fuel are limited. Where will we get the biomass to create liquid fuel? We have to realize that most alternative energy sources are focused on production of electricity. So the need for a source of liquid fuel is very demanding, both in the development of the resources to produce the cellulosic biomass to convert, but also technologies to convert that into liquid fuel.
We're going to need something that we can put into a gas tank or into an airplane engine. While there are other different sources, wind and solar, what we need is liquid fuel, and biofuels hopefully can do this.
It's pretty clear that petroleum is going to come to an end. The use of petroleum, at least on the scale it's now used, experts disagree as to the time scale over which this will occur. Obviously, we have domestic concerns involving how much oil we have to import. There are also global climate change concerns as regards CO2 emissions. So I think the reason why he would place so much emphasis on biofuels is the possibility of it being a viable alternative to petroleum and/or fossil fuels in general.
Switchgrass was noticed by the Department of Energy in 1991 as a possible resource for cellulosic biomass because it's indigenous to North America. It grows everywhere from Maine out to the Rockies and from Minnesota down to Florida. And it produces a lot of biomass per acre. And it also is acclimated to marginal soils. Therefore, it won't compete for arable land that we use as a food resource.
Well, switchgrass is just one of those things that grows everywhere. I mean, you can't kill it no matter how hard you try. So it's everywhere. It grows like crazy. I mean, I can attest just showing you guys how many times I cut this stuff back before it pops back up again. So, I mean, it's a huge source of biomass. It's all easily converted into fuels. Well, I wouldn't say easily converted into fuels. Grows on marginal land you'd never use for crop development. It's got a natural herbicide resistance, pest tolerance. It's perennial. It requires low inputs of nutrients. So there's a lot of reasons why switchgrass would be the best choice.
Switchgrass seems to be one of the candidate plants for ethanol development. It seems with the biomass that you get from the plant, you can get more ethanol out of that when that comes about. Switchgrass is native to this country, so we have plenty of areas that we can grow the switchgrass. It seems to be a good plant to work with, unlike some of the other ones.
Switchgrass, for instance, is a perennial plant that does not have a long history of, in particular, biotechnological investigation. And so we're working on improving the sorts of things you need to do from a biotechnical approach to improve those processes, to make possible the genetic modification of switchgrass, for instance. It's not exactly like baking a pie. Some of the ingredients are, while they exist, they are not highly developed.
One very important aspect of our research here is developing methods for creating a strategy for gene confinement. We all realize that transgenics, that is the ability to genetically modify all of the different perennial plants that would be useful for biofuels, is going to require a robust gene confinement strategy. And what do I mean by that? That we need to make plants that are sterile so that the genes that we introduce are not communicated to wild populations. And given that these are all perennial plants, this is a requirement. And so that to create male and female sterile lines will give us the ability to, one, create hybrid plants that will be higher yielding, but also provide a strategy for gene confinement that once we add genes to the genomes of these plants, we know that they will be contained out in the environment. There are a lot of other cellulosic sources that people are looking at. So switchgrass is not really a magic bullet. It's one of many different cellulosic materials that can be developed into liquid fuel. For example, there are other plants like miscanthus is a grass, pennisetum, that all produce a lot of biomass per hectare. Wood products like poplar, eucalyptus, willow trees, all of which can produce a lot of biomass per year, per acre, that can be converted into liquid fuel. So switchgrass is a leading contender for crops that we can produce, but it's not the only one. And the technologies that can be developed for converting it into liquid fuel will utilize any of those cellulosic sources.
The number one obstacle is simply cost. If you can make a product that costs less than gasoline and produces somewhere in the neighborhood of as much energy, people will adopt it. Or better yet, if you can make something that's cheaper then you have a good chance of convincing people to convert to that form of energy.
It's the breaking of the cellulose into the sugar molecules to make your ethanol. I think that's where the hang-up is cellulose is a pretty tough compound, and how we can efficiently break that molecule apart is our hurdle right now. We don't want to be adding heat with propane or anything like that, which is going to bring our costs up. We want to be able to extract the cellulose or break the cellulose bond in order at a very cheaply in order to bring the costs down, and I think that's where we'll start to gain in the development.
I'm sure there's a lot of money to be made. I mean, agriculture is big business. You know, frequently people have the image of the farmer with a cow and some chickens and a pig and the tractor, and that does exist and there's nothing wrong with it, but something on this scale would be pretty industrial, I would imagine, and it would be worth, yeah, many millions and millions, perhaps billions of dollars.
When you do this type of work, it's inevitable that you will develop new techniques as needed. That's part of the basis of science in general. And you will develop new techniques that are applicable to agriculture in general. So the development of a robust gene confinement strategy for switchgrass has also applications in developing hybrid plants and rice. So the same genes are involved, and developing these techniques, I think, is more broadly applied than switchgrass, although that is our primary focus right now. And you know, I should add that we are also looking at different trait genes that we would introduce into switchgrass and other plants. For example, the ability to move genes into these plants is like a big erector set, where if we think of a trait that would be important to biofuels development that is conferred by a gene, we have the ability to test out whether or not that gene would be significant in switchgrass for biofuels. The same sort of procedure can be applied to other crop plants. For example, we're funded here on a separate project to develop corn varieties that are nutritionally improved for germplasm in Africa. That project focuses on increasing carotenoids, the precursor for vitamin A, as well as increasing iron levels in maize varieties that would be useful in Africa. So the ability that we have to move genes into plants, express them, and evaluate their outcome allows us the flexibility to create and evaluate things that would be useful through agriculture to people in general.
You know, I don't have my Superman outfit on, but I think I could contribute towards the agricultural portion of things. As my academic career expanded through all the years that I was in academia, I learned that using genetic engineering and molecular techniques could be a way in the future to help more people, because we could take genes from other places and put them into the plants that could alleviate some of this hunger. Golden rice, vitamin A, and rice is a good example. A lot of people only eat rice, so we can't give them, you know, they may not eat a banana. So what is the nutritional value of bananas? Can we take some of that nutritional value and put it into rice? And that's what Ingo Petrikis did with rice and vitamin A, but he took it from daffodil. So we can use throughout the kingdom genes that can help us with hunger. It gets me up in the morning. Yeah, I think it does contribute to things. And some of the things that we've done in the past 10, 12 years, we've helped answer some questions that biotechnology has wondered about. We've worked with transgene escape, and we have ways that we can alleviate some of these problems that arise making transgenics. So although there are questions and concerns with making transgenic crops, there are also answers to those concerns, and this field can address those.
In genetics, you can't very much work with people. You can say, well, let me find some volunteers and make mutants from people. say this guy will be defective in this gene and this guy will be defective in that gene, and we'll put them in these glass like receptacles and see how they think. That's immoral. Can't really do that. So they would use something like a mouse, which is 88% genetically identical to a human. And so you use a model. So a lot of people know a lot about how the mouse works, but they extend that to say, well, if it works in a mouse, it pretty much works the same way in person. And they do the same thing with plants. They use another model plant. And so we're all have a common ancestor if you go back far enough. You could use the information that I can get and extend that to other organisms.
The final product you come up with makes it all worthwhile, you know, that you're hitting milestones to achieve a larger goal in the bigger scheme of things, you know, nationwide, globally, you know, and just for the human race in general.
There are a lot of intelligent people in power, but the thing is that holds them back isn't necessarily the rationality that comes from an argument based on the scientific data of how this would improve things. I think that if you ask an individual straight out what the stakes are, and then like you just show them this is really what it, no one is making up this data, this is really what it says, I think a lot of people would be concerned and would be convinced and say, well, you know what, science has done great things for society, and anyone who thinks it hasn't, I guess just don't get sick. Don't get sick and just, you know, experience the idea that I can rub some sort of antimicrobial on my skin and suddenly like a rash goes away. I think people are smart enough to know if they're just told straight up really what's going on. I don't want to be a pessimist and think that they just will never care.
We still have a long way to go. Right now, breeding and molecular trait development in switchgrass is a beginning. We still have to be able to refine the cellulosic ethanol out of the plant, and that seems to be a hurdle right now. But I think over time, I think we'll get over those hurdles and possibly, you know, get making more fuel for less cost.
I think biofuels are a necessity for the future generations. It doesn't take you too far to look to see that the fossil fuels we've been using now are causing a lot of problems and will not be there forever. There is a finite amount and it will eventually run out. So we need to start emphasizing a renewable and sustainable energy choice. I would say that switchgrass or biofuels in general will be one of those upcoming platform technologies. I mean, it'll start getting us away from relying on foreign countries to provide energy for us. It's also going to be beneficial to the environment, and you don't have to look much further beyond the recent BP oil spill to see how detrimental some of these fossil fuels can be to, you know, the economy and the environment.
I think inevitably, it is essential. To call it alternative fuels is a misnomer right now, as if that were a luxury item. And we've relaxed a little bit about our approach to this, it seems, in this country, given that the price of oil has come down. But a recent issue in Science did a review on scaling up for alternative energies. And there is some, finally, great information that's been published about what resources we have left in terms of fossil fuels and how much time is left. Not only fossil fuels, but different energy sources. And what you see there is that we've got a limited time left on oil. We have a little bit longer on coal. And natural gas, we have some resources there, but still it's finite. And after that, what happens? You know, how long will it take? Well, I don't think we have time to sit on our hands and wait for that to happen and then say, oh my goodness, we have to develop, you know, an alternative source for fuels. And when you look at it, wind, waves, solar, nuclear, they all make electricity. How are you going to make liquid fuel, which will run our cars, our boats, jets, tanks? You know, it becomes almost a matter of national security. You know, that we have to have a renewable and sustainable source of fuel that does not contribute to the carbon cycle.
It's going to be a combination. I don't think it's going to replace oil. I think we're too far into oil to really back out of it. It could start to alleviate some of the pressure that we see when there are problems with the oil. I think there are many uses for ethanol, and refining it out of cellulose, one of the most abundant compounds on this planet, we can tap a resource that we haven't been able to tap before.
I mean, I like to think that it's a lot prettier than fields of oil rigs and stuff like that set up across the countryside. Yeah, I do. I think that you could, you know, obviously it would alter the landscape slightly, but it might be in a better way.
I think that this is critical. This is critical research being done. We're not going to be able to burn petroleum forever.
To convince people in power that we need to do something about climate change is a big debate. What drives the public is the price at the pump. There's no question about that. You know, that when the price of gas was nearing $4 a gallon, and I told people what we were doing here, they were like, why don't you just get back in the lab and keep working? Now, the situation is a little bit more relaxed, but that is misleading, because obviously oil is finite. What will you do? So I think if the price of oil were to go back to $4 a gallon, people would become a lot more supportive of this type of research. In fact, why don't we just make it $8 a gallon? And we'll use the extra money to support research. We have to get this done. We have to get this done. I don't know, it's like I said, it's not an alternative. I don't view it that way.